The overall goal of this procedure is to use an image analysis platform to rapidly analyze and explore fluorescent microscopy images. This is accomplished by first seeding cells into a plate, adding perturbations to each well, and incubating the plate. Images are then acquired using an automated fluorescence microscope, and the images are semi-automatically loaded onto the PhenoRipper image analysis platform using the PhenoLoader tool. After setting the analysis parameters, PhenoRipper automatically identifies recurrent block types in the images and generates phenotypic profiles for each image that can be explored using the Pheno browser. Ultimately, the final clustergram shows relationships between different conditions and image features, allowing for the comparison of complex yet subtle phenotypes generated by the high-throughput imaging screen.
The main advantage of this technique over existing methods that require automated identification of individual cells is that it is much faster and easier to use.
This method can help answer key questions in the biomedical field, such as how different drugs affect cellular processes.
This procedure begins with cell culture and cell fixing as described in the text protocol. Stain the fixed cells using the specific primary antibodies, fluorescently labeled secondary antibodies, and other fluorescent stains that can be found in the text protocol.